Hi, my name is Marissa Fuss, and this is my Advanced Concepts Presentation 3. Okay, so to begin, here's some background information. Cholesterol doesn't contain long hydrocarbon chains, which is a feature that many other lipid molecules have and are ultimately known for. Cholesterol is also a more well-known lipid molecule in the medical field because of its negative relationship with the human heart. On the other hand, cholesterol is used in the generation of particular steroid hormones, some of which include testosterone, estradiol, progesterone, and cortisol. Another use of cholesterol is in the cell membranes of animal cells. Within these cells, cholesterol is important in ensuring that the membranes' properties are regulated in order for them to carry out their functions in the body. Most generation of cholesterol occurs in the liver, even though it can be generated in other tissues of the body. Generation of cholesterol has a direct relationship with the amount of cholesterol that is present in the body at any given time. So then moving forward to the biosynthesis of cholesterol. In a three-step process, cholesterol ultimately receives the 27 carbon atoms in its structure from acetyl-CoA. The generation of isopentyl pyrophosphate is the first step in the process. The generation of squalene is the second step in the process. And the generation of cholesterol is the third step in the process. Location of each of these three steps is important to note. The first step in the process occurs in the cytoplasm, whereas the second and third steps occur in the endoplasmic reticulum. More specifically, these steps are in a portion of the endoplasmic reticulum that is commonly referred to as the lumen. So now, step 1. Step 1 in the process starts with the generation of isopentyl pyrophosphate using acetyl-CoA. Acetyl-CoA and isoacetyl-CoA are first used in order to generate 3-hydroxy-3-methylglutarol-CoA, or HMG-CoA, which is right here to the right. Then you have mevanillinate, is then generated using HMG-CoA reductase, which you see right here, and then this would be the final product. This generation is referred to as the committed step in the overall process. The reason for this is because this step is the process that is the main control point in the generation of cholesterol. This concept will be explained more later on in this presentation. Moving forward to the second half of step 1. Step 1 in the process continues to ultimately generate 3-isopentyl pyrophosphate. This generation occurs in a series of 3 reactions. These reactions utilize ATP in order for them to be completed, which is seen right into the figure to the right. It is important to realize that isopentyl pyrophosphate is commonly referred to as being the activated 5-carbon isoprene unit. This generation of 3-isopentyl pyrophosphate is what concludes the first step in the generation of cholesterol. So now step 2. Step 2 in the process begins when isopentyl pyrophosphate isomerizes to ultimately become dimethyl pyrophosphate, which is seen right here. One of each C5 units are ultimately used in order to successfully generate squalene. Squalene is considered to be a C30 isoprenoid. The actual reaction that is completed in order to generate squalene will be examined more on the following slide. So now, the generation of squalene occurs in this following set of reactions. 2 molecules of isopentyl pyrophosphate, which is right here and right here, and 1 molecule of dimethyl pyrophosphate, which is right here, are used to generate phosphonyl, which is right here. Then you have 2 molecules of phosphonyl are then used to combine to form squalene. The generation of squalene is what concludes the second step of cholesterol formation. So then you see the whole overall process to the figure to the right. Then moving forward to step 3. Step 3 is the one that ultimately generates cholesterol. Squalene epoxide is the first to be generated from squalene with the help of O2 and NADPH. So you have squalene, and then it goes to squalene epoxide. And then an intermediate is generated before lanoestrol can be generated, which you see right here is the intermediate, which then forms lanoestrol. Okay, so then the intermediate ultimately rearranges in order to generate the lanoestrol, which is right here. And then in a 19-step process, you have the formation of cholesterol. So more specifically, lanoestrol is generated when squalene epoxide is cyclic sizes. Then you have the removal of carbons, also, as I said before, occurs in a 19-step process. And then from there, after all these steps are taken, the formation of cholesterol is performed. Okay, so now how lovastatin inhibits high cholesterol. Compounds that are commonly referred to as statins can be used in order to inhibit high cholesterol. The way in which this is completed is through inhibiting the generation of cholesterol. One example of this type of compound is lovastatin. Another name for lovastatin is Mevacor. The explanation of how these how lovastatin works will be included on the next slide. So then you have an example of, here's the lovastatin molecule, and then here's melvinate, which was explained previously. Okay, so compounds like lovastatin are considered to be competitive inhibitors. They are involved in preventing the HMG-CoA reductase from carrying out its function. As previously mentioned, the function of HMG-CoA reductase is an extremely important section in the overall process of generating cholesterol. Cholesterol levels in a person's body can be reduced by approximately 50% when lovastatin and bile salt reabsorption inhibitors are given to him or her. This could potentially be helpful in lowering cholesterol levels to patients who suffer from atherosclerosis. Overall, compounds like lovastatin are effective in preventing people from having high cholesterols in their body. So then here would be my works cited, and that would be the end of my presentation.